really important, even though some people take it for granted, is that really there's no society so far that has been shown not to have developed a musical tradition. So music is universal. Every society across the world has, in some form, music. And what makes it even more interesting is that there are some forms, some uses of music that are shared across cultures. And these uses, these functions, often entail social interaction. For example, gathering, music associated with being together with other people. Healing, some person trying to make another person feel better through music. Or infant-caregiver interaction, the typical observation that lullabies and play songs are used throughout the world to regulate the mood of our infants, our pre-verbal infants. And so to put together essentially what we know is that archaeological, developmental, ethnomusicological evidence points to musicality as a universal trait in humans. And what I try to do with my research is to understand why, how, what for. That's what I try to do in my lab. So you may already have noticed how I turned the language here from music to musicality. Okay, what is musicality? So music, you can think of it as a cultural product, something that we can create with our own creativity and depending also on our history, our experience and so on. Musicality is something different. Musicality is the biological predisposition that makes humans potentially able to perceive, to make, to appreciate music. And this is something I think is in part universal. Some of what makes us capable of making and listening and enjoying music is shared across individuals. Something else probably is not. But there is probably a core, common code, which is biologically grounded, that makes us all musical. And I investigate the biological origins of this capacity, such musicality, its development, so how it develops throughout life, and the role it plays specifically in social interaction. Specifically because I'm interested in social interaction, I don't consider music only as sound, but I also consider it as action, movement. And the latter is really important if you want to study social interaction, because the sensory encoding of music ends between your ears. Like you listen to it, and there is no possibility for an observer to understand what's going on in the back of your mind. Action instead, it's something that allows us to create a sort of window into our state. Depending on how I move, my face, my body, my limbs, people can infer a lot of what's going on within me. And this can become a potential bridge, connecting people, tuning people in the context of social interaction, including, for example, through body synchronization, something that occurs both when we make music with someone else, but also when we listen to music together with other people. Synchrony, movement synchrony. Now, I was happy to be part of this event, specifically because this event talks about the methodological challenges and perspectives. So, I think that to investigate the kind of theoretical topics I presented so far, we do actually need to advance our techniques considerably, because we are really ill-prepared to study this phenomena in the lab properly. When I began studying neuroscience of music, the majority of the sensory stimuli that were presented in a lab sounded like this, beep, and the majority of the motor action that you would study in the lab looked like this. Okay, so yeah, okay, that's for sure it's necessary to start simple. This, I take it for granted, but I think that recently there's been a very great advancement of computational models that we can use to try gradually to see how it's like to study music when it happens in its real environment. So, real music that we listen to, or real musical movements that we perform. And this is what all the studies I will present are about. They are an attempt to getting closer and closer to real musical activities, real musical performance. Okay, first sip, sorry. Okay, so the presentation is organized across a number of keywords that I think we have to consider as a sort of starting point to innovate the methodologies that we have to study musicality. And I'm going to start with behavior. So why behavior? Well, because behavior, it's kind of fundamental, but it's really poorly studied. In neuroscience, we pay very little attention to behavior. Often there are studies that ignore it altogether, like they study the brain as if it is a sort of separate organism from the world. But I mean, behavior is how we survive, and the brain, in fact, is mostly developed to control behavior, because if you don't do that, you die, and your species ends with you. So it's crucial to study it. And truth is that even if people often in neuroscience diminish the importance of it because they consider it kind of simple, it's not true. The behavior is actually extremely complicated to study because it's multidimensional, it's rich. It's really hard to study properly in the lab. I make an example, right? So these are beautiful examples of coordinated dynamics that occur across different taxa, right? So as humans, we can dance with other people and synchronize our movements, but you see this phenomena even in birds and fish and so on. And one thing that struck me is, how can we do that? Like, it's really complicated and also beautiful to watch. You can come up with different accounts. Now imagine that these two dots on the screen are two humans, but they could be anything else, and they move together at the same time, kind of in a coherent manner, and us as observers would all agree, oh, they are synchronized. Yes, of course they're synchronized, but how do they reach such synchronization? Now one account would propose, oh yeah, because they're mutually adapting to each other, like one is following the other, and therefore they are in sync. True, possible. But what if, yeah, there was just a response to a shared common input, right? Think about fireflies. They like to respond to light. Crickets do that too. If you throw a bit of food in the water, a lot of fish come and they kind of synchronize. Later there will be probably a buffet. You will synchronize to that too. Okay, so I mean, and this is interesting. These two are not necessarily alternative, but they are certainly a different way to explain how we synchronize with others. And now the nice thing of music is that specifically in the context of dance, well, actually this is happening at the same time. Because if you dance with your partner, wherever, in a discotheque, at home, and so on, you can achieve synchrony by simply paying attention to the music without even looking what the other is doing. Or if you actually pay attention to what the other is doing, you will follow the movement of your partner. And everyone who can dance can tell you that it's really crucial to pay attention to what your partner does. So they do happen at the same time. Okay, now, Felix Bigon, a postdoc in my laboratory, wanted to basically compare these two accounts, really in the context of dyadic dance. And focusing only on behavior, what we did is we invited a number of laypersons, so people without specific dance training, okay, laymen people, to the laboratory. And we played them music that would make them dance, but we didn't ask them explicitly to dance. People were recruited in dyads, in pairs. We asked them to just look towards the other and enjoy the music, to do whatever you want. We are measuring your movement, so they figure maybe movement is important, but they didn't have to move, they were not supposed to move. Of course, putting music that is made to make you move ended up making them move, right? And the music sounded pretty much like this. One example, well, we have 8 different tracks. Okay, most of you recognized it, right? Yeah, so it's Michael Jackson, Thriller, and yeah, there were different versions of this. And now, what do we do experimentally? Well, on the one hand, to tease apart to what extent we need music to synchronize with the other, we had them play the same music to both, which they receive with the earphones, okay, in fact, in their ear. So either the same music or different music, slightly desynchronized music. So occasionally you will be dancing with a partner, like in a silent disco, where your partner is actually dancing to different beats, okay? Or we allow them to see each other, so they could potentially adapt to the movement of the partner, or not, because there was a screen in between, okay? So it's a 2x2 manipulation, where we permit audio to drive the synchrony, yes or no, same different, or visual contact to drive the synchrony, yes or no. Okay, now let's start from the data. We recorded motion capture with landmarks around the body, like we had, I think, 30 of them. It's a motion capture Vicon technology. Are you familiar with this? Yes. Okay. And how often have you encountered someone who collected a huge data set with Vicon, like 50 markers, 100 participants, and then in the end in the paper, they show only one marker on the head that does this, okay? So to me, like, having been in this for about 20 years, very often. And the truth is that it's because, as I said in the beginning, behavior is complex. It's not easy to analyze. We don't have a lot of methods either. And so here we came up with an idea that it's very simple computationally and very effective. To reduce the dimensionality, the dimensionality complexity of this data, we use PCA. So we submitted the movement trajectories of the different markers for each of the dancers to PCA to extract 15 principal movements, which are very reminiscent of common dance moves. I'll show you that in a moment. And so this is quite great, because from a lot of different markers on the body and the X, Y, Z representation of where they are over time, you kind of break down the dimensionality substantially, going into just 15 principal movements that are also interpretable. Like instead of just the head that moves this way or this way, you know that, I don't know, it can be like head bob or something else. I'll show you how it looks. So on the left, an original representation of a subject dancing to the music. And then one by one, you will see how this can be broken down into these different principal movements that are occurring at the same time. So this is side sway, vertical hands, twist with your hands, or front, back sway, side hands. There's also twist, bounce, side shuffle, head bob. If you ask someone who's used to dance, they will tell you that these are common dance moves. It's stuff that you can see in the discotheque, in isolation as well as combined. The great thing here is that they're happening together, combined, but we can disentangle them and study them one by one, individually, okay? Yeah, I like to present it over and over because I like a lot this video. Alright, so let's go to the results. First I summarize the fundamental result. Now, what did we find? Looking at each movement and how well each movement synchronizes across people as a function of listening to the same music or being able or not being able to see the other, we found something that was quite unexpected. We found that there are some movements that synchronize through vision of the partner, and then there is a different group of movement that synchronize through the presentation of the same music. But there are no movements that synchronize through both additively. So these two drivers of synchrony do not interact. They occur in parallel. Okay, this is quite interesting and striking, because it means that if I can see you and you're listening to a different music, even though your movements will be partially desynchronized to mine because your music is faster or slower, turns out that I will still synchronize with some movements with you, even if you're actually moving at a different beat. And at the same time, some other movements will synchronize to the music. Now, first I tell you which these movements are, and then I explain you how this is possible. Okay, the movements that synchronize through vision of your partner are side sway, twist with hands, side hands, and side shuffle. Instead, the movements that synchronize through the music are vertical hands, front back sway, upper body rock, twist, and head bob. Okay, cool, but how? That's a tricky thing. How can you do that? Well, actually, it turns out that it's thanks to a spatial organization. So the movements that synchronize through vision are all movements that essentially take advantage of this dimension, the X dimension, they are lateral movements. And these are the ones that you basically can't resist synchronizing with your partner, and I tell you why, because they're very salient. Like if I move in this way and you're facing me, this will be a very salient signal, okay? And this is a signal that you synchronize no matter whether your music is in sync or not. However, at the same time, other movements on the anterior-posterior dimension, so movements that are more like this, and they're more difficult to be decoded by your partner because they are hidden from the view of your partner facing you, but those continue synchronizing to the music no matter what. And now, in this kind of dichotomy, there is only one movement that sort of behaves in a different way, that is special, and this movement is bounce. Now, bounce is the only movement that synchronizes more both when I see you and when we see the same music, but still not additively. So these two things don't happen together. They are still two main effects without an interaction. And there are a few other things about bounce that we found very interesting. So, for example, it's the only movement enhanced in amplitude when I see my partner, potentially because it's working like a signal, a cue. It's also the only one that is synchronized to the musical beat. Here you see basically how strongly synchronization is as a function of the musical beat of the music. Here the beat will be 1 on the x-axis, and then you have 2 times the beat, 4 times the beat, which would be the musical bar. And you can see that only one movement, PM10, which is bounce, is tightly synchronized to the beat. So they really bounce in time with the beat. All the others synchronize with different periodical levels of the music. And that's why we hypothesize maybe bounce could be seen as a sort of supermodal pitch setter, something that you can use to synchronize no matter in which modality, an hypothesis that we might test in the future. Now, if you're wondering how these all look like, now I have a representation of the movements that synchronize with the partner, no matter what the music is doing, and that's the movement in orange. Now you have together all the movements that were synchronizing with your partner. In green, you have the movements that instead synchronize with the music, no matter what the partner is doing. And finally, bounce, what we call hybrid, because it kind of doesn't really fall orange or green, it's something in between. So we call it hybrid synchronization. Okay, so this was behavior, right? But I said at the start that I am a cognitive neuroscientist. So for me, it's interesting also to study the brain. But I don't want to study the brain in isolation. I want to ideally study the brain and behavior together, holistically, which is also something that is kind of neglected, because there's attention on one and the other. But as I said in the beginning, in neuroscience studies, behavior is really often taken on the corner, so to say. And so now I'm going to give an example of a study where we try to integrate brain and behavioral sciences at the same time. Okay, so this is a study that was led by Dr. Atish Kul, another postdoc in my lab. And so, and this also has to deal with synchrony. But now, it's not synchrony about our bodies, it's synchrony about our brain activities. And I'm lucky that the talks that preceded mine have already discussed this, right? This so-called hyperscanning community, measuring more than one brain when people do things together to see to what extent these brains exhibit signals that codepend on one another, okay? And that one way to show that is to show that they synchronize, and they indeed do synchronize, because now there are tens and tens of papers that show how, in the context of a social interaction, brains do synchronize. Turns out that we don't know a lot about how these emerge in the first place. For example, we don't know what are the minimal conditions that are necessary for two brains to synchronize. And this is what Atish wanted to figure, okay? He said, how do they spontaneously synchronize? How does it emerge in the first place? And so we thought, okay, how about getting rid of a task? So simplifying the social interaction as much as possible. We invited people in the lab. We asked them to sit facing each other. We measure a lot of different indices of behavior and brain. I'll tell you later about that. And we said, just look towards your partner, okay? It's awkward. Look at the feet, okay? Behave spontaneously, don't speak, don't make gestures, and that's it, okay? We're gonna measure this. And then we're gonna manipulate a number of things. On the one hand, whether we can or cannot see our partner. So sometimes I have my partner sitting in front of me, some other times there's a screen, so I cannot see that. And we also try to manipulate proximity. Are we far, 3 meters away, or close, 1 meter away? Proximity didn't yell anything, okay, spoiler. But visual contact did. I'll show that later. And then meanwhile, we have EEG from both. We have video cameras to reconstruct spontaneous body movements from both. And we have eye tracking to see where people look at in real time. Okay, first thing, synchrony. We looked at synchrony by looking at the correlation between the envelope of neural oscillations in delta, theta, alpha, beta, gamma. Okay, this is a very slow signal that you can extract from EEG. Like the first talk today discussed EEG mostly as ERP related or very fast signals related, true. But when you look at neural oscillations, you can also extract other features from the signal, such as the envelope, which is instead very slow. It kind of falls more in a time scale similar to F-nears. And in fact, it also correlates sometimes with F-nears. So now we're taking these very slow signals across the brains of the people, we look into what extent they're synchronized, and we find out that in some parts of the brain and in some frequencies, so in the occipital and the tempoparietal parts of the brain, in the beta and in the gamma oscillations, indeed, when people can see each other, well, their brains are more synchronized. And this is interesting on its own, because I'm showing you that just seeing the behavior of a person in front of me is sufficient to somehow synchronize the two brains. However, it's not really explanatory, right? It's a kind of descriptive result that you don't really understand because you wonder how. And that's why we need to measure behavior, right? How? Well, you measure behavior, you find out. And we did measure behavior, so we can find out. So what did we do with behavior? Now, we crossed together the video recordings, which were submitted to a convolutional neural network, which was able to reconstruct the position of different body parts in the space of the video camera. You can do it for the face, so you can reconstruct the position, for example, of your eyes, your nose, your mouth, but you can do it also for the whole body, so you get information about the limbs, the trunk, and so on. And then you can cross that with the eye tracker of the person in front of you, so that you can also see, given a certain movement in a certain time, where were people looking at, okay? You can really kind of figure out the interaction history throughout time. And it looks like this. So what you see is 3 videos of a subject in front of another subject, and then so the blue dot is where the subject is looking on the top, and the red one is the center of the face, and then the time series is telling you how close to the face is the subject looking at. So you have information in real time that tells you to what extent we're kind of doing eye contact. So how close to your face I'm looking now, and now, and now. And you have information about how much I look at your eyes, essentially. How much people are moving over time, spontaneously, you know? These are more postural adjustments, things that you would do in this awkward experiment where you face another person you know, but you cannot say much about. And also smile, like by computing the distance between the lip ends, we were able to reconstruct how much people smiled at each other in real time. The beauty of this is that it not only is data-driven, but it's also spontaneous behavior. Right? We're not asking them to do any of that. They do it. They create the data with their spontaneous interaction. So it's closer to what happened in real life, much closer. And when we cross this information across participants, we find something really cool. We find that these social cues, so to say, so eye contact, body movement, smile, they too synchronize interpersonally. So if I look at you, you're likely to look back at me. If I spontaneously move, you're likely to move at the same time. And if I smile, you're likely to smile back. So behavior also synchronizes spontaneously between people, okay? So this brings us to the situation where we have different brain activities that synchronize, but also different behaviors that synchronize. And the question the obvious question that people ask me normally is, okay, but what comes first? What codes what? Okay, what codes what? We cannot test with EEG. Causality and brain activity is something that you have to do with different methods. But what comes first, this you can do. Because, for example, you can assess an index of instantaneous synchronization. So you can create a time series that tell you at any given point how synchronized the behaviors or the brain were. And then by using either cross-correlation or Granger causality, you can estimate to what extent one anticipates the other, such as if our behaviors get synchronized at time 0, our brains are likely to be more synchronized at time 1, or vice versa. Okay? Now, let's go for hands. Who think that the brain activity, synchronous brain activity, anticipate behavior? Raise your hand. Okay, who think that the okay, no, first. Okay, brain synchrony predict behavioral synchrony. Raise your hand. Let's try. Who think that we first synchronize our brains and then we synchronize our behaviors? Who would predict that? It's a guess. It doesn't have to be correct. Okay, and then who think it would be the opposite? Like, yeah, exactly, and that's exactly what happens. So we see that some behaviors, especially the spontaneous body movement and smiling, anticipate the emergence of synchronized brain activity, especially the cluster that we find in the occipital cortex in gamma. And Granger causality supports the same conclusion too. Okay, now we take a deep breath. I take a sip of water. Because the next point is called computational. So how this science has to also develop new computational methods to integrate behavior and brain activity. Now please recall the structure of the first experiment that I presented, the one where the people were dancing together. Now in aging, how we replicate everything, but we include EEG from both. Okay, now that's, how many of you have worked with EEG in the past or have been trained to use electrophysiology in the past? Okay, some people. Now, what I recall from my classes when I was a student, it was like, don't do it, you cannot measure people moving with the EEG because there are too many artifacts. And that's true, like, it's true because muscular activity on your face and your neck will leak in the EEG, and muscular activity is kind of an order, if not 2 orders of magnitudes, larger than the activity elicited by the brain, which makes it really complicated to extract any real neuroscience measure. However, in the past, there's been the development of a lot of methods that allow you to separate these 2 sources of information. For one of such, which we particularly like and which we have validated in my lab as well, is called artifact subspace reconstruction. Also based on principal component analysis, essentially the algorithm is able in real time, even if you want, we didn't do it real time, but you can, to separate the raw data from what is called pre-processed data in orange. And then you can basically see how this can transform very nicely artifacts that are injected in your data, such as these blinks, that are this kind of activities that happen when the subject is blinking or moving the mouth, and returning you an EEG signal that is, of course, not perfectly, but decently decontaminated by this activity. And you can do that across a bunch of movements, such as movement of your head, movement of your hands, swinging, of course, and they say, this we didn't test though, even jumping. Okay. Now, our subjects, they don't jump, but they did kind of move, like, you know, they sway, they move their head, they move their hands. So the first thing is, we use this to denoise it a bit, but that's not sufficient. It's a start. We denoise it. The real challenge here is that in the context of dyadic dance, there are a lot of things that are happening at the same time. And the challenge is, how can we separate these different processes that are important for listening to music and at the same time controlling your movement, motor control, and then at the same time monitoring what your partner is doing, and then also at the same time coordinating your own movements with the movements of the other. Like, it's hard to argue that you're not doing all these things when you're dancing with someone, and I think it's also hard to argue that these things are not in part independent from one another. And neuroscientifically they are, because there are different networks that orchestrate all this. And the challenge is to find a method to decode them when they occur at the same time. Okay? Okay. To do that, we use a method that is called temporal response function. Okay, so what does this do? Okay, imagine that the brain is a black box. It's a mysterious machine that receives an input and produces an output, but you cannot do anything about what it's doing. You don't have this information because it's a black box. So you have the output, which is the EEG, and you have the input, which is a feature of a signal. Let's pretend for now that the signal is just an auditory signal, so an acoustic recording, the one that I'm plotting on the left. So what you do is very simple. So you take the input, the output, and then you use a linear convolution to basically model the relationship between the two. Okay? This leads you to identifying what are called temporal response functions, which essentially tell you how, given a change in the input, there will be a certain response in the output. And the beauty of this method is that if you try to do that with brain activity listening to speech or music, you find temporal response functions that are actually extremely similar to very established neurophysiological responses to sound onsets. So these responses that you find to model your EEG are very similar to the responses that people normally measure in the lab when they do the boring experiments such as beep, sorry for the word beep pop, beep pop. Now it's beautiful because it allows you to exploit a lot of well-known neurophysiological research to actually study not only the response to your beep, but now the response to a very complex stimulus from the beginning until the very end. And you can do that in the audio, but you can do it also in the video dimension. In fact, it's also done with videos. Okay? Now here, to make it even more complicated, we did something that is called multivariate temporal response function, because we don't want to only predict how the brain is tracking the music. We are simultaneously trying to predict brain activity also based on how the body is moving. We have this information because we use the Vicon to record it. We also want to predict how the brain is responding to the movement of the other person. And we have this information as well, because we recorded it. And we even want to see whether, to some extent, the brain is tracking the degree to which I am in synchrony with you. So we also have a representation, the fourth time series on the plot, in pink, that tells you to what extent you tend to be in phase or in antiphase in your movements. Here we focused on bounce, so essentially the time series is telling you whether you're bouncing in phase with your partner or you tend to bounce more in antiphase, under the assumption that these are very distinct, right? Bouncing in antiphase gives you the sense of being out of sync. Bouncing in phase will give you more the sense of being in sync. And on top of that, in the same model, we also include recordings of the muscles, because we know that even though you remove some of it with the pre-processing, some of it will be there for sure. Or in any case, you cannot be sure. So you want to record it. And we recorded the neck, the face, the eyes, and include those as well. Okay, so now you're creating this huge, mastodontic, multivariate regressor where you will be able eventually to see how each of these individual predictors explain unique variants of the EEG. By unique variants means that it explains something on top of all the other predictors. Okay, so where my movement is more predictive of me, of the brain activity, compared to your movement, the music, the muscular artifacts, and so on. And this is a representation of how it works. So you can imagine events occurring in the music or in the behavior on the left, triggering a representation of how the brain activity would look like based on the temporal response functions, right? So you have an event and then you're modeling your EEG. Eventually, once you have created a model of the entire EEG based on all the predictors, you basically confirm how similar this EEG is to the real EEG, okay? And in this way, you can essentially separate these contributors. But let's look at how the data looks like. Let's see. Okay, so this is how the brain responds to music. And it responds very similarly to music across conditions, with a morphology that is very compatible with what we know from neurophysiological studies, the beep-like studies. It's called, there are a number of responses called like P50, N100, P200. The neuroscience experts will know what I'm talking about. This is fairly comparable across conditions. And so is the tagging of the movement controlled on your body. This is very similar of the readiness response potential. It's also a measure related to the control of movement. If you zoom into different types of movements, so movement of your left hand, right hand, your feet, or your head, you are even able to separate that. So you can even selectively extract index that is specifically related to the control of a different part of your body. Now we go into more interesting ones. So for example, we find indeed the predictor that uniquely track the movement of your partner in the occipital cortex, which makes sense in the visual system. So there is some activity that is uniquely explained by what your partner is doing, and it's there only if you can see your partner when you have visual contact in those conditions. And then we found also something really interesting. We find that the brain is also tracking synchronization. So there is also a response that tracks to what extent your movements are in sync with your partner. And it's also occipital, so it also probably has to deal with visual processing. And I think it's a beautiful example of a sort of complex phenomenon that emerge from the interaction of two simpler phenomena. Okay, what do I mean here? To see this response, it's not necessary, it's not sufficient to test an individual alone or the observation of another individual alone. This is an emergent property of two people that are doing something together, and when you rule out the activity that explains how you move or the activity that explains the monitoring of how your partner moves, there is something left, and this is the interaction between the two. So essentially it's a response that you cannot test and you cannot measure unless you put two people together doing something together, which tells us even more convincingly that we really need to turn to real life scenarios and ecological conditions, because otherwise there are some phenomena that the brain is able to do that we cannot really study. And this is while ruling out activity from the muscles. So these are the predictors of the eyes, the cheeks, and the necks. These are all ruled out from our model. Now, to speed up, I will go a bit faster. You might recall how I said how bounce is somehow special, and all these special features. Well, now we do something with bounce more. We try also to predict how well each different type of movement predicts brain activity. And we found that bounce is the best predictor of brain activity, both of the movement that you perform, as well as the movement that you observe, which reinforces this idea that bounce is probably a very salient signal that captures our attention better than other type of movements that are produced simultaneously. Okay, believe it or not, I will go on and change even more. And now I'm going to talk about comparative research. Comparative, in this case, means comparing different species. Why? You might recall this slide at the beginning when I said that music is a bit everywhere, and to me, a possible explanation of why music is so widespread is that maybe music exploits some very, very old neural system that we share with other species, that we might have inherited by a different species that is not there anymore. Okay? Again, that would not mean that everything we do musical relate to these mysterious species that preceded us, no? But that there might be some fundamental capacities that we have, such as the capacity to encode fundamental features of music, I don't know, rhythm or melody, that we might actually have inherited. So we had already the toolkit, and then we learned how to use it to create and invent music. And this is a question that we try to address in our lab by comparing how adult humans, newborn humans, less than 48 hours old, and macaques respond brain-wise while listening to music. Okay? When we started this, they told us that we were crazy because we played Bach to the monkey. And now I'm going to play you an example of the many stimuli that we have played to our macaques. So this is an example of Bach chorale. It's monophonic piano. And it goes on for about 2 minutes, and there are another 9. And then we have a control condition. It's a shuffle condition where we shuffle the order of the notes as well as the order of the intervals, okay, to kind of break down the temporal and melodic structure altogether. And they sound like this. Now, I have friends that study contemporary music. A friend of mine is also a composer. His hero is Schoenberg, and to him this is music, and I'm fine with that. And it's okay, but this to me is a cultural consideration. Like, I think when you study musicality, like the predisposition, it's hard that you consider this music. Like if you ask a 2-year-old, for example, I don't think they will say it's music. In any case, it's a good control, right? And we measure EEG from all these groups. And we analyze the data in the same way I told you before, using this temporal response function, which now comes in very handy, because what we can do is we can use some kind of low-level representation of the music, such as when you have an onset in the music, a note, or the spectral flux, which give you an indication of how much a spectrum is changing over time, but also a higher-level representation of the music, a representation of surprise, which you can assess using a model, for example, the one by Marcus Pearce, which is able to tell you, given a certain note and the past history of the music, how surprising that note is, both with respect to the time, as well as respect to the pitch, okay? So you can create a representation of how surprising all the notes are by having essentially a model that learn on a lot of music that has been produced by humans over the course of history. And now what we do is we plot basically how well we can predict the activity of the brain of the macaques and of the humans by using either only the acoustic representation, which is the dashed line that you see below, and then also when you include these higher level representations, such as the pitch in orange, the timing in green, or both of them together in black. And the first striking thing is that try to ignore the orange and green for now, focus only on the black. So first of all, we can actually predict the brain activity of the macaque using surprise, okay? Which implies that to some extent the macaque is actually able to make expectations about how the music will unfold. These macaques have never listened to music their entire life, but they seem to be equipped to produce some kind of expectations. But when you break down the contribution of the pitch and the timing, you find that this result is entirely driven by the timing, right? The macaque is able to make predictions about the when notes occur, but not which kind of note, while the adult human is able to predict based on both. So the human listens to music in a different way, anticipating how the timing will unfold and also how the melody will unfold, while the macaque is kind of deaf to the melodic structure, but is able to anticipate timing. Now, the $1 million question here, unless you have read our most recent paper, is how will the newborns behave? And again, I want to ask for a vote. I ask for a vote because I think it's interesting to see how sometimes our predictions are confirmed or not confirmed by the actual data, because it gives us a justification for doing curiosity-driven research. Okay, so how many of you think that the newborn will behave more like the adult humans? And we have a bunch of hands. And how many instead think they will behave more like the macaque? More hands. And the right answer is newborns behave more like the macaque. So time-wise, they are already wired in, but melody-wise, no. So based on this, it's only one experiment. I will hypothesize that the melody tracking emerged later, probably as a result to exposure to speech, music, and so on. We're trying to test this directly in the lab right now. This is another analysis of the same data using ERP that shows the same thing. I'm not going to go into it because I don't have time, but you can ask me later. Because before I close, do I have a bit more time? 10 minutes, perfect. Before I close, I want to move into a second type of comparative research that we conduct, which doesn't deal anymore with the phylogenesis, so to say, so how we inherited traits from our ancestors throughout evolution, but on the ontogenesis, which is how humans develop musical traits throughout the ontogenetic development. And this is something that we do with our colleague Qin Yuan, who wondered how does musicality emerge? When? Sorry, not how. When does it emerge? And by musicality here we mean both sensory, so tracking of the music, but also motor, spontaneous movement, engagement in response to the music. So to do that, we invited a bunch of infants to a laboratory in Vienna, where we're doing this work in collaboration with the developmental lab. And the infants were 3, 6 or 12 months old, and we also have an adult control group. We measure their brain, EEG activity, but we also measure how they move, and we do that with the video recording and then a reconstruction of the movement, like we did in the second study I presented. I played you an example of one of the play songs that we presented to the infants and to the adults. So that's one. I know it's very simple, but they're also very young, so you need to do something that is appropriate. Again, we have a shuffle control, like for the macaque study, we shuffle the position of the notes and of the intervals, and you get something like this. It's actually musically interesting, I agree. In the ears of the young fellows, you'll see. Okay, so that's our control condition. And then now we start from the brain, okay? Now, of course, you would expect the human brain to track better the music compared to the shuffle. This has been showed by many people, including us with the newborns, in fact. And when you epoch your EEG to each of the tones comprised in the music, you find a very nice auditory response, which is much larger than the response elicited by the shuffle music, okay? So that's kind of a benchmark that we use, okay? But now you go back, like backwards in ontogenetic development, and essentially you find this response across all the groups, right? Which is, again, not too surprising, because I've just shown you how newborns are able to make predictions about timing. Probably these infants are as well. One thing I like very much, though, is how we captured the development over time of these physiological responses, which become faster in their latency and larger in amplitude. This is also a known phenomenon in neurophysiology, but this is beautiful because you can really see how it progressively gets faster and faster. Now let's move to movement. Okay, now we are looking at how these infants are moving spontaneously while listening to the music. And now we're using again the principal component analysis as a tool to reduce the dimensionality of the data, which give us, in this case, 10 principal movements that explain a bit less than 80% of the entire kinematic variance. And here you have a bunch of movements. Remember, the infant was sitting on a high chair, so they can't jump, for example. There are things that they cannot do, but it was mandatory to do it this way because 3 months old cannot do much else but sitting, and 1-year-old can do a lot of other things, but if you want to compare them, you need to put them in a comparable context. And here you get front back rocking, side sway, a sort of proto-clapping, leg kickings, and so on. And now what I show you is how much these movements are expressed across different age groups, as well as a function of the experimental conditions. And here we find a very nice effect, which is that only the 1-year-old, the 12 months old, move more spontaneously in response to music compared to shuffle, while the 3 and the 6, they don't. Okay, that's interesting, because now earlier I told you how the brain is already equipped to decode information from the music, right? But when it comes to responding to it, moving the body accordingly, well, this seems to occur much later, because at least in the first year you don't find much, and then at 12 they seem to move more when they listen to music. And this is true for some movements in particular, such as front-back rocking, side sway, proto-clapping, up-down rocking, and arm pedaling. But now we dig a bit more, because we're not happy enough. We want to understand better how this is going. And so what we do is we reconstruct how the music looked like with an acoustic envelope, and how the movement looks like, putting together all the principal movements. We have these 2 time series that tell you how the music is unfolding, how the movement is unfolding, and now you want to see the relationship between the two. And to do that we use Granger causality, which is a technique to see how much the history of one variable predict the future of the other. I want to see what is driving what, essentially, okay? And the first thing I find, which is important, is that in general, music is a better predictor of movement than movement of music, and that's significantly so, which makes sense, but it's important to demonstrate, really, that music and how it unfolds explain better how movement will be later than the other way around. But when you compare it across different conditions and you look at it as a function of the lag, so which means how much back in history I have to go to see how you will move in the future, you see that especially around 200 milliseconds there's a top kind of peak prediction accuracy, which means that essentially there's kind of a buffer information, optimally 200 milliseconds, that predict the movement of the infant in the future, and this is true for all the age groups. So even though only the 1-year-old is moving more in terms of amplitude, when it comes to trying to predict whether it's moving or not moving in the future, well, that you can already do. So it means that there is a sort of very shallow, very simple, we call coarse coupling between the music and the movement at 3 months, but it's not as sophisticated as what happened at 1, where they really move more in response to one compared to the other. We also wonder whether, for example, given a specific event in the music, there will be a specific response in the movement, such as I hear a beat and I produce a movement. We don't find evidence of that, and we don't find evidence of synchronization either. We looked also at whether the periodicity of the movement was similar to the periodicity of the music. None of that is happening. So that has to develop later. In fact, some people suggest that it developed in late childhood. Okay, so movement sophistication develops with age. Alright, so now I'm done. I will just have a very few slides. Do I have 2 minutes? 2, perfect. To talk about the last topic, which will introduce what Sarah Balde, PhD student in my lab, will be talking about. And this leads to making music inclusive. Okay, what do I mean by making music inclusive? So I did my PhD looking at the brain of musicians, okay, and how they interacted with one another. My dream was to test this kind of stuff in everybody, because I think that there's a lot about musical interaction that can be generalized to any sort of interaction. So to me it's an excellent model of coordination and communication. But not everybody agrees with that. And the reason is that you can test musical interaction mostly if you study musicians. But musicians are kind of very sophisticated machines that have been trained over the course of time, and it's difficult to generalize from this sometimes elite population to the overall population. And that's why when I was in Sydney at the Marx Institute, I developed this device which is called e-music box, which is essentially a carry-on, like the old style music box that many of us I'm sure have had when they were in their childhood, but just a bit more sophisticated because well, we 3D printed it, because it works with the software in synergy with the computer, because you can make it play any music you want, and you can manipulate other features of the music any way you want. And also because it gives you beautiful data, so like suddenly the music becomes an oscillator, and of course this resonates with my interest into synchronization and so on. Of course it's kind of transversal, you can use it to study people across different age, across different neurodevelopmental conditions, because it's very simple, as a function of different cognitive abilities, musical training, and even cultures. So now I'll give you a very quick overview about the kind of studies that we're doing using this tool. So for example, one of such, following on the work with infants, is an attempt now not only to see how the baby is moving spontaneously when listening to the music, this is from the data that I showed you earlier, but how it changes if it's mom that is playing the music for you and is able to actually see how you respond to it in real time. Which I'm a parent of 2, okay, and like, I think this is really a thing. Like, you use music to communicate with your infants, and you track how they respond to drive their arousal, for example. But it's not very well studied, and this is a tool that you can use to get insights into this. And this data we are analyzing, so I don't have yet much, but more autism, right? We know from a lot of literature, from the very beginning, from the first description of autism, from Leo Kanner in the 40s, I think, that people with ASD are highly interested in music, okay? More than any other conventional communication, but we don't have many tools to make music accessible to them from a motoric perspective, okay? So when it comes to teach them a song, yeah, okay, but what about something like this, that it's really fun and can be adapted to please their needs? And so along to machine in my lab, I developed a game that people with ASD are playing from home, synchronizing with their caregiver, and so they kind of control these two planets orbiting around the sun, and the two parts that they control, each control one part, like more rhythmic or more melodic, and the music become more loud when they achieve synchronization. So it's a way to use music as leverage, as a platform, simple platform where people can meet and interact socially, under the hope that this can promote social behavior on a larger scale. Okay, so it's kind of an intervention study, if you might. Okay, so in the past we have shown how cognitive abilities modulate our capacity to jointly make music, even in non-musicians. For example, this is a study from a few years back where we show that people who are higher in empathic perspective taking, it's kind of a measure the everyday tendency to get into someone else's shoes, right? If you score high on this measure, you're also better at synchronizing with someone else when you make music, which tells us something about what are the cognitive mechanisms that support joint music making. And finally, like comparing joint music making across musicians and non-musicians, we're very interested in this phenomenon where when people play together, you have movement, and I said the movement for me is a fundamental way to share and to coordinate and synchronize, but there are different kind of movements, right? There's an instrumental movement that you need to make the music, and that's actually similar to the output you hear sometimes. But there's another one that's called ancillary, which you don't need to make music. Like if a piano player moves the head like this, I mean, this is not becoming sound, this is vision, this is for the eyes. And people look at this stuff, right? And this guy is looking. And so we wonder, can we first of all try to create models to separate these type of movements that happen at the same time? Or are they different? How are they different? Do they support synchronization? Like the capacity to see your movement, the ancillary of the instrumental, does it help me actually to synchronize with you? And does any of these require musical training? So this is a phenomenon that you can study in everyone, so musicality, or it actually requires some training, more like music. So if you're interested in the answer to these questions, well, you need to wait until 3:30 when Sana will provide you the answers. And that's it for my talk. Thank you very much for